She asked if all the answers were the study guide were easy to find in the notes, and I said, do not these answers for this study guide.
No, I haven't given you the answers for any of the study guides, just the practice exams.
Oh, yeah.
But the answers are taken. So what I do is I look at the slide and I write the questions. So I just go with the slides. Go down. Okay, so those are the phases. And then the next column is labeled chromosomes. Okay. What are the chromosomes doing in interphase? Yeah, they're unwound. So what does it look like? You could draw a little picture next to it.
Jungle.
Yeah. Okay, so that's interphase. I put unwound. Okay, so they're being used. There's enzymes and other things we'll talk about when we talk about replication and transcription, translation. Or transcription at least. Okay. How about in prophase? What are they starting to do? They're starting to condense. Okay. Okay, what does that look like?
More towards the center.
Right, so, and you have light and dark areas. Okay. What happens in pro-metaphase? It's more condensing to where they're linear structures. What does it say on there?
It says attaching to microtubules.
Okay, so the envelope dissipates, and now you have microtubules attaching. Yeah, so I took the nuclear envelope off because that dissipated. And now you have these structures, the microtubules from the centrioles, and they're attaching to the chromosomes. And then they're going to start pulling them. And what happens in metaphase? Yeah. They align or meet in the middle. At what structure? Metaphase plate, yeah. So then, and how do they align? Top of each other or side by side? Top of each other, all right. Okay, and then these guys are attached. And then what happens in anaphase? They pull apart. If you want to use, so another thing you can do is use the first letter to help you remember what it's doing. They meet in metaphase. You could say they are preparing in prophase and prometaphase. They meet, they pull apart. So this looks like
So pro-metaphase, they're not X's.
they are. I just good observation. Okay. In telophase, yeah, they keep. Actually, they make a new. This one's kind of hard. They make a new envelope. And now they're in. And they start to make two different cells. So, I mean, you can say they tear. What does the thing say? Okay. Tear away two opposite ends. All right, and then cytokinesis, you have two separate cells. What's this structure that forms during cytokinesis? The cleavage furrow. Good. All right, what's the next? Does it have cytokinesis? The next one is the nuclei. Okay, and we drew what the nuclei were going on here. The nuclei are dark here. You have dark and light areas there. Nope. Yeah, I've shifted one. They're dark in interphase. Dark and light. In prophase, and then it dissolves in prometaphase. It's still gone in this one, still gone in this one, and then it reforms in telophase. Right? How that takes place. All right, what's our next one?
Other characters.
Okay, so that's the last one, right?
I would say what it basically is happening.
Right, so this describes what's going on. What does it say for other characters? For cytokinesis? What's happening during what's happening during interphase?
Cell grows and duplicates organelles.
Right, so you have cell growth and duplicate organelles. Prophase, what does it say?
Spindle apparatus assembles.
Okay, the spindle apparatus. What's the spindle apparatus? That's these microtubules, these asters, that pull the chromosomes to the opposite side. What does it say for prometaphase? Congregation? What's congregation? Alright, so yeah, they get together, go to the center. What does it say for metaphase? Nothing? Okay. Because, I mean, we basically described everything that goes on in metaphase. It just comes in a little more progression. In anaphase?
Cohesion proteins remove A.
Okay. Cohesion proteins are removed. What are the cohesion proteins? No. Right, they hold them together. So that's what's holding them together in metaphase. They're removed in anaphase, and so they can pull apart.
What are they called?
Cohesion proteins?
Is it like a centromere?
Yeah, they're at the centromere.
Okay.
They're what form those. And then you have two types of anaphase. Anaphase A and B. What's anaphase A? Right, so you have the pulling of the chromosomes to opposite sides. What's anaphase B? The actual cells themselves pulling farther apart. All right, and then in telophase, what's the telophase? So then we don't need the spindle apparatus anymore. that it? Okay, and if you wanted to write for cytokinesis, you could put cleavage furrow is formed, separate the cell.
I have a question.
Yeah.
When does telophase end and cytokinesis begin?
Cytokinesis begins with the formation of the cleavage furrow. Telophase ends when the nucleus reforms and the spindle apparatus is.
Okay, so like, okay,
Disassembled.
I was thinking like it ended when the cells, like just when the cells split apart. But you're saying that happens in cytokinesis?
Well, they are separate. They're separate processes. So telophase is when you get these chromosomes, the spindle apparatus is no longer there, it disassembles, and then you get a nucleus reforming. Cytokinesis is the process by which the cells split.
Okay.
So telophase only has to do with the chromosome and the nucleus. Cytokinesis has to do with the cells themselves, the forming between the cells. That's a good question. This will be on the test. Okay? So no, and it will help you in other parts of the test. If you know this information, then when you look at different nuclei, you'll be able to tell what's what.
Okay, what was the thing in the middle called cleavage furrow?
Yeah.
That's only in animals, right?
Right. So what happens in plants? They don't have a cleavage
Cell
furrow. They have a cell plate. Why don't they have a cleavage furrow?
They don't have a membrane.
They do have a membrane. They do have a plasma membrane.
Okay.
But they also have, you're on the right path, but they also have a cell wall. So if they just split their cytoplasms, they'd be separate, but they need that cell wall in there too. So the cell plate is actually what divides the cell, not the cleavage furrow. Okay, I'm going to pull up some pictures of some nuclei and all right, I don't know. That's working out. Okay, there will be some pictures of some nuclei as well. So you're going to have to be able to look at the pictures and tell what's going on. Okay, so that's what I'm going to go over right now. Stages of meiosis, fading in. Mitosis, thank you. So if you take, you looked at the onion epidermal cells today, right? What did you see?
I couldn't see that.
Hopefully you saw cells going through different stages of mitosis. Okay? Maybe it looks something like this. Gross. This one is stained a little better, so you can see the nuclei, you can see the chromosomes probably better, and it's much more close-up view. So, what is going on here? What do the nuclei look like? Jumble, but you can see dark and light spaces.
It's in prophase.
So it's in prophase. Good. Okay, what's going on here? They're lined up in the middle, right? Metaphase. Okay. What's going on here? Separating. So anaphase. Tearing apart. All right, now what's this one? Yeah, telophase and maybe a little bit of cytokinesis going on there, but yeah. Okay. These might be a little harder. You can see them a little bit. All right, so these two. Okay, what's this one? Prophase, right? You can see the light and the dark. This one, is interphase. Now let me see what's going on. Okay.
How's that interface? I can see white.
No.
Well, yeah, it's more interface than that one. Okay, maybe this one's interface. Yeah, you can't see it very well. But some you get the idea. This is what? Interface. Well, this is anaphase. You can have different stages. So here you can see nuclei are reforming. So that's telophase. This is a little bit later. You're starting to see the cleavage furrow there, cytokinesis. All right. This is probably early anaphase. This is later anaphase. They're just starting to pull apart here, or what Michael would call anatelophase. Right? Okay. All right. This is from an onion cell, so this is probably similarly to what you should have seen under the microscope. What stage would this be? Prophase. Okay, so you can clearly see you got dark and light areas. What about this one where it's all pretty much black? Interphase. Okay, good. What about this one? Metaphase. Kind of have to use your imagination a little bit. Yeah, no, that's about all I see. I don't see any anaphases.
When I tell you I just use my imagination.
I was going to say, you use your imagination wrong on this one. I will give you some clear ones, so it's not ambiguous. But, I mean, this is just showing that you have a bunch of cells that are going through different stages of mitosis at the same time. Okay. Why an onion root? Why do you think we get the slide is of the onion root? Right, so that's where you get a lot of growth in the ground. Okay, so things are going through mitosis. All right, here's some cool ones, I think, and they're labeled. Okay, so it's easy. Interphase here. Look at that nucleus. Can't really see anything. Prophase. All right. Here, these green dots are what? Yeah, the spindle apparatus is forming. Okay. And it's attaching to these different chromosomes. Okay. So that'd be prometaphase. This is of a cell. I'm not sure exactly what kind. An animal cell. In metaphase, they're all lined here in the middle. Okay, they don't look perfect like the drawings that we make, but they're still. Anaphase, you can see they're pulling apart. They're going opposite ends of the cell. And then telophase, they've completed and they started to make their own little nuclei. All right, so you got it? All right. Number your page 1 through 6. And these are not the same. I shifted them around. Okay. 1, 2, 3, 4, 5, 6. Wait. Let me give them back to you on this. Take it on this. Take it on the same piece of paper. No, but I don't want to have you can erase your name. I'm writing it. Yeah, I'm just handing out. I don't even care if it's yours. Just taking the top one and handing it back. One, 2, 3, 4, 5, 6. Okay, real quick, I'm going to stop recording. Yes.